So we have now mitosis. Mitosis is a type of cell division in which a cell divides to form two daughter cells retaining the same and equal chromosome number content. 44XY becomes 44XY and 44XY, and this is called as mitosis. The chromosome number remains the same, and there is equal distribution of genetic material. So the mother cell and the daughter cells are having same chromosome content and are genetically the same. If the two daughter cells by themselves become mother cells, they will form exactly the same identical organism. If you remember, when zygote forms to form two blastomeres, and both blastomeres are produced by mitosis. And if by mistake both blastomeres separate, they will form identical twins. In other words, mitosis may both daughter cells are genetically the same as that of the mother cell. The chromosome number is the same, quantity is the same, and quality of chromosomes, genes are also the same. And in this process, division of nucleus and cytoplasm takes place only once. एक ही बार न्यूक्लियर डिवीजन और साइटोप्लाज्मिक डिवीजन होता है. Meiosis, it took place twice. Two meiosis, meiosis 1, meiosis 2. Mitosis is only once. Mitosis is a common type of cell division which is responsible for growth and repairing of wear and tear in multicellular organisms. Why does mitosis take place in us? Zygote से बेबी और बेबी से हम बड़े होते हैं तब तक कंटिन्यूस माइटोसिस होता है. हमें कोई इंजरी हुई, फ्रैक्चर हुआ, स्किन पे टेयर हुआ, तो रिपेयरिंग द वेयर एंड टेयर, हीलिंग द फ्रैक्चर, all takes place by mitosis. In eukaryotes, in unicellular organism prokaryotes, you may have amitosis. Some prokaryotes have mitosis. One cell will divide to form two cells. That means reproduction has taken place. तो उससे reproduction होता है. Mitosis helps in increasing the number of that organism itself by reproduction. And since this division results in the formation of somatic cell, it is called somatic cell division. Where does mitosis take place? In your body cells. Body cells are called as somatic cells, and hence it is called as somatic cell division. Here in mitosis, it maintains the chromosome number in the daughter cells equal to that of the mother cell. So it is called as equational division. The number of chromosomes in a daughter is equal to that of the mother, hence it is called equational division. And in animal cells, there are two distinct centrosomes, and the kinetic spindle fiber is organized with reference to centrioles, hence called centric. Animal cells में ऐसा होता है और प्लांट सेल्स में ऐसा होता है. एनिमल सेल में सेल मेम्ब्रेन तो होती है, बट देयर इज नो सेल वॉल. सेल वॉल नहीं होता है. सेल वॉल इज एब्सेंट इन एन एनिमल सेल. तो ये है सेंट्रियोल्स और ये है सेंट्रियोल, और सेंट्रियोल टू सेंट्रियोल स्पिंडल फाइबर बनते हैं. Hence, it is called as centric. In plant cells, ये है animal cell. In plant cells, you are having a cell wall and spindle fibers arranged from cell wall to cell wall. तो उसमें centrioles नहीं होते हैं. No centrioles are involved, and spindle fibers arise from cell wall to cell wall. So animal cells में the centrioles and centrosomes Our spindle fibers are organized with reference to centriole, hence such a division is called centric. That is called centric. But in plant cells, there is no centriole, hence it is called as acentric. Where do spindle fibers arise from? Cell wall to cell wall. The term mitosis was called by Walter Fleming, a German biologist who observed it for the first time in 1878. So Fleming is credited with the discovery of mitosis. Chalo, all this was very simple today. Let's now get into a beautiful part of the chapter. How does it take place? Cell division of mitosis takes place in two steps: karyokinesis and cytokinesis. The division of nucleus is called karyokinesis, and division of cytoplasm is called cytokinesis. So you have nuclear division and cytoplasmic division. Nuclear division called karyokinesis and cytoplasmic division called cytokinesis. सबसे पहले karyokinesis, nuclear division. In the S phase of the interphase, the DNA, which we remember is called chromatin thread, replicates to form two daughter DNAs. And daughter DNAs get condensed to form chromosomes. DNAs का chromosome बनता है. Beautiful point. I'll explain you soon. The daughter chromosome separate by karyokinesis, so each daughter cell gets identical and equal number of chromosomes. This is very interesting. ये मजा आएगा. Karyokinesis or nuclear division is a stage in which nucleus undergoes a series of changes to form daughter nuclei. And what changes will take place? Four phases: prophase, metaphase, anaphase and telophase. ये चार phases होते हैं in which you will have nuclear division called karyokinesis. सबसे पहले let's see prophase. Prophase is the longest phase. सबसे लंबा phase prophase होता है, and it is for purposes of convenience divided into early prophase and late prophase. In the early prophase, ध्यान से, chromatin network condenses and resolves into definite number of chromosomes. Chromatin network becomes chromosomes. Initially, each chromosome appears as a single stranded monad chromosome, thin and long. And as condensation progresses, each of them reveals two distinct chromatids held together by a centromere, which is called as a dyad chromosome. ये dyad chromosome बनता है, and chromosomes undergo dehydration and coiling to become thick and short. तो डीएनए कंडेंस होके क्रोमोसोम बनता है बाय डिहाइड्रेशन एंड कंटिन्यूअस कॉइलिंग. ये आपके एनसीईआरटी में दिया नहीं है, नोट्स में भी दिया नहीं है. आई एम शोइंग यू दिस इन अ लिटिल मोर डिटेल. ये ध्यान से सुनना, किसी भी बुक में दिया नहीं है. सर, here we have maternal DNA and here we have paternal DNA. Where did paternal DNA come from? The sperm. And where did maternal DNA come from? The egg. So this is, say, the first cell, which is the zygote. And N is maternal chromosome. And the other N, blue, is maternal chromosome. Black is paternal chromosome. How many threads of chromatin are there? We have one maternal thread and we have one paternal thread. So it is N maternal, C one chromatin thread. N paternal, C one paternal chromatin thread. So the cell is 2N, 2C. What does 2N represent? N maternal, N paternal. N maternal, N paternal. So it is 2N. And what does 2C represent? One chromatin thread maternal, one chromatin thread maternal, and one chromatin thread paternal. So it is 2N, 2C. In the S phase, the DNA replicates. So one maternal thread, blue, which is double stranded. DNA we all know is double stranded. The two DNA strands will separate. ये आ गया आपका ब्लू थ्रेड और उसके साथ आ गया a new red thread which is genetically the same. So this has now become blue and red, and the other blue thread also becomes blue and red. So two blue threads have formed blue red, blue red. Similarly, black thread, paternal DNA, double stranded DNA, becomes black yellow, and that means one maternal thread has now become two maternal threads, and one paternal thread, black, has become two paternal threads, and that is called DNA replication. So, how many N's are we having? N is N maternal and N paternal. Paternal, maternal remain same, N. But how many chromatin threads are we having? We are having maternal chromatin thread, maternal chromatin thread, paternal chromatin thread, paternal chromatin thread. So 2C has now become 4C. 2C has now become 4C. This was 2N 2C. 2N 2C. In the zygote, after S phase, it has become 2N 4C. This is N2C. This is N2C. This is N maternal 2C threads. This is N paternal 2C threads. So N maternal 2C threads, N paternal 2C threads, two chromatin threads. So it has become 2N 4C. एंड एक्चुअली ये तो एस फेज था. नाउ हाउ इज एक्चुअली दिस हैपेंड? इफ यू रिमेंबर, डीएनए अकेला नहीं होता है. डीएनए के ऊपर छोटे छोटे बीड्स होते हैं विच आर कॉल्ड एज हिस्टोन प्रोटीन्स. सो डीएनए हैज विथ इट छोटे छोटे प्रोटीन्स विच आर कॉल्ड एज हिस्टोन प्रोटीन्स. And here we have. So we have histone proteins. Histone proteins, उसके ऊपर होता है डीएनए. हिस्टोन प्रोटीन के ऊपर होता है डीएनए. हिस्टोन प्रोटीन के ऊपर होता है डीएनए. हिस्टोन प्रोटीन के ऊपर होता है डीएनए. And actually you have something called as a beads on a string appearance. Beads on a string. So this blue thread is actually DNA histone, DNA histone, DNA histone, DNA histone, DNA histone, DNA histone, and that is called as a chromatin fiber. So ये है chromatin fiber. So ये है maternal DNA NC. ये एनसी था ना, maternal DNA. It is actually maternal DNA NC in the form of maternal chromatin fiber. Similarly, bead string, bead string, bead string, bead string, bead string is paternal DNA, which is NC. तो ये था आपका एक्चुअल जाइगोट. जाइगोट में ये था. And in the S phase, the DNA undergoes replication, and NC will now become N2C, and NC, your NC will become N2C. So N2C, N2C, will become 2n4c. N2C, N2C will become 2n4c. तो यहां पर भी NC, NC S phase में one bead on string will become two beads on string. If you remember, DNA replicates, but in the S phase, along with DNA, histones also replicate. DNA के साथ साथ histone भी replicate होता है. So we have N2, NC becoming N2C. N2C. तो ये क्या हो गया? N2C maternal. This has become N2C maternal. Similarly, NC paternal has now become N2C paternal. N2C paternal. So N2C, N2C has become 2N4C. Has become 2N4C. ये कौन से फेज में होता है? S phase. S phase. From S phase, the cell will go into early prophase. Early prophase में what will happen? ये bead string, 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 bead string. First, all the beads start coming closer to each other. So it will now become condensed by dehydration. Look here. Bead string, bead string, bead string, bead string will undergo condensation by dehydration and will become bead, 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 bead. तो bead, 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 string, 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 string. So bead string, bead string, bead string will now become bead, 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 string, 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 string. And that means once again, bead string, bead string, bead string, bead string, bead string, bead string, bead string will now become bead, 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 and string, 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 string. So the length of the chromatin thread will condense. Condensation of the chromatin thread takes place, and that bead structure is called as a nucleosome fiber. What is that called as? A nucleosome fiber. So bead string, bead string, bead string, bead string has now become bead, 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 string, 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 string. And that is called nucleosome fiber. That will happen to both maternal threads, and that is N2C, and both paternal threads. So that will become N2C. So you having N2C, N2C. N2C, N2C. What are such fibers called as? Nucleosome fiber. The bead nucleosome fiber will keep on coiling. How will it coil now? अभी bead, ये देखना ध्यान से, bead will now become bead, 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 like a telephone wire. तो समझो आपके पास एक स्ट्रेट वायर है, उसको आपने टेलीफोन वायर जैसा कॉइल किया, तो लेंथ ऑफ द टेलीफोन वायर विल डिक्रीज. या समझो एक टेलीफोन वायर को आपने इलोंगेट किया, लेंथ ऑफ द वायर विल इंक्रीज. वो पुराने जमाने में, अभी भी टेलीफोन होते ना विद वायर, टेलीफोन वायर वाले. हाँ, आजकल तो कॉर्डलेस और मोबाइल्स हो गए हैं, बट पुराने जमाने का जो वायर टेलीफोन वायर जैसा होता है, तो ये है वायर, टेलीफोन वायर. एंड समझो एक लंबा सा फाइबर आपने टेलीफोन वायर जैसा कंडेंस किया. सो bead, 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 bead will now become, look at here, bead, 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 and the length will become further condensed. That is called as a solenoid fiber. So, nucleosome fiber has become solenoid fiber. And solenoid fiber, beat, 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 like a telephone wire, will coil again. So it will now look like, यहाँ देखना, beat, 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 beat. So it has actually become beat, 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 beat. So beat, 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 beat has become beat, 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 beat. So length has further condensed, and beat, 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 beat solenoid has now become beat, 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 beat is called super solenoid. And super solenoid will further condense, will now become beat, 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 beat. And super solenoid condenses to form super solenoid and will keep on condensing till it forms bead, 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 and यहाँ पर भी bead, 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 and this is one unit which is called as a chromatid. So entire bead has now become bead and has become chromatid. And chromatid और यहां बन जाता है another chromatid which is called as its sister chromatid. If you remember, this one mother DNA had become two daughter DNAs which are genetically the same. So, both these chromatin threads are genetically the same. So both these nucleosome fibers are genetically the same. So both these solenoids are genetically the same. So both these chromatids are called as genetically the same and are called as sister chromatids. And two sister chromatids form one chromosome. What do two sister chromatids form? A chromosome. And one chromosome is formed consisting of two sister chromatids. Two sister chromatids ban jate hai. So, what has DNA become? Bead string, bead string, bead string has become two bead string, bead string, bead string in the S phase, which has then become bead, 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 string, 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 nucleosome fiber, which is then like a telephone wire become a fiber called as a solenoid fiber, which has become super solenoid and further coiled to now form units which are highly condensed and are called as chromatids. And chromatid नाम के दो स्ट्रक्चर्स बन जाते हैं continuous dehydration and coiling. Dehydration and coiling. And two chromatins has become two chromatids. Two chromatin has become two chromatids. Initially, both chromatids are long and they look like a single structure. and are called as monad chromosome. Initially, each chromosome appears as a single stranded monad chromosome, and ये है monad chromosome. It undergoes further dehydration and coiling, and the two chromatids become visible and different. तो पहले मोनाड बनता है which is identical and looks like one. With further dehydration and coiling, it appears distinctly two. So two chromatids appear, which is now called as a dyad chromosome. So monad chromosome now becomes a dyad chromosome. One becomes two. And in a dyad chromosome, two structures are important. ये है एक dyad chromosome. In the center is a structure called as the centromere. तो दो क्रोमैटिड्स के बीच में एक सेंट्रोमियर होता है. What is present between two chromatids? Between two chromatids is found, यह दो क्रोमैटिड, उनके बीच में एक स्ट्रक्चर होता है called as centromere, and that is called as a centromere. And on the sides are found proteins. On the sides are found proteins which are called as kinetochores. तो दो प्रोटीन है, बीच में जो प्रोटीन है व्हाट इज दैट कॉल्ड एज अ सेंट्रोमियर. बीच वाला प्रोटीन इज कॉल्ड एज अ सेंट्रोमियर. एंड साइड में जो प्रोटीन्स है व्हाट आर दो साइड प्रोटीन्स कॉल्ड एज काइनेटोकोर्स. साइड प्रोटीन्स आर कॉल्ड एज काइनेटोकोर. काइनेटोकोर से अराइज होते हैं स्पिंडल फाइबर्स. तो क्रोमोसोम जब अरेंज हो जाएंगे, काइनेटोकोर से अराइज होंगे स्पिंडल फाइबर. काइनेटोकोर से अराइज होंगे स्पिंडल फाइबर. सो दिस इज वन maternal chromosome joined by centromere made of two chromatids. Similarly, these are two chromatids. These are two chromatids and joined by centromere with proteins called kinetochores on the side, and this is a paternal chromosome. तो ये वाला कौन सा हो गया? मैटरनल क्रोमोसोम. ब्लू वाला मैटरनल, ब्लू इफ यू रिमेंबर वाज मैटरनल. और ब्लैक वाला कौन सा हो गया? ब्लैक वाला इज अ पैटर्नल क्रोमोसोम. तो मैटरनल and paternal chromosomes are formed, and what is this phase called as? Early prophase. This is called early prophase. How many such chromosomes are formed? We have 23 pairs of chromatin threads. 23 maternal chromatin threads and 23 paternal chromatin threads. 23 maternal and 23 paternal chromatin threads. So, how many pairs of 2 and 4C are formed? 23 maternal and 23 paternal. So, how many how many chromatin fibers are there? 23 pairs. 23 pairs. How many nucleosome fibers are there? 23 pair. 23 maternal, 23 paternal. So how many chromosomes will be formed? Number of chromosome formed will be 23 pairs. 23 maternal chromosomes and 23 paternal chromosome. 23 maternal chromosome and 23 paternal chromosomes. So 23 maternal and 23 paternal, that makes our chromosome number 46, जो हमने reproduction में भी किया था. So we have 46 chromosome, 23 maternal and 23 paternal. In what form are these chromosomes now formed? Dyad chromosomes. ये कौन से chromosomes है? Dyad chromosome. What was this phase called as? Early prophase. This was called early prophase. Then we have late prophase. In the late prophase, the nuclear membrane and the nucleolus begin to disappear. The daughter centrioles in animal cells start moving away from each other till they occupy the polar position. तो ये है अर्ली प्रोफेज. अर्ली प्रोफेज के चेंजेस किए. लेट प्रोफेज में क्या होगा? लेट प्रोफेज में ये जो न्यूक्लियर मेम्ब्रेन है, न्यूक्लियर मेम्ब्रेन स्टार्ट्स डिसअपियरिंग एंड सेंट्रियोल्स जो बीच में थे, centrioles were in the center. Now centrioles start moving towards the opposite poles. Centrioles start moving towards the opposite pole. तो न्यूक्लियर मेम्ब्रेन एंड बीच में होता है न्यूक्लियोलस. करेंगे हम लोग एक दिन. न्यूक्लियस में होता है डार्क एरिया कॉल्ड न्यूक्लियोलस, which is concerned with protein synthesis. स्कूल में शायद सुना होगा. Nucleolus begins to disappear. Nucleolus begins to disappear. Nuclear envelope begins to disappear. So nucleus ही disappear हो जाएगा. And centrioles start moving towards opposite poles. Centrioles move towards opposite pole. What is this phase called as? Late prophase. So early prophase में DNA और chromatin thread condenses to form dyad chromosomes. How many of them? 23 paternal and 23 maternal. ये condensation and dehydration से 23 maternal, 23 paternal dyad chromosome बनेंगे. That is in early prophase. Late prophase में nuclear envelope disappears, nucleolus disappears, and centrioles move towards the opposite poles. This is prophase. Then the cell goes into metaphase. In metaphase, condensation of the chromosome is completed and thick chromosome get arranged along the equatorial plane. Hence, the stage is called metaphase. Meta, middle phase. This phase is characterized by formation of kinetic spindle. Spindle is made up of proteinaceous chromosomal fibers and interpolar fibers, which is called mitotic apparatus. In animals, spindle fibers radiate as an aster from the centriole and is called astral rays or astral mitosis. In plant cells, asters are absent, hence it is called anastral mitosis. Now, what do you mean by all this? This sounded a little tough. So, sir, diagrammatically, what will happen in the metaphase? Metaphase में chromosomes completely condense हो जाते हैं. तो monad chromosome had become dyad in the early prophase. Dyad further condensation करते हैं and will become distinct chromatids joined by centromere. तो ये distinct chromatids आ जाते हैं which are joined by centromere. So distinct chromatids joined by centromere. And where do they get arranged along the equator? तो ये इक्वेटर में अरेंज हो जाते हैं. कौन कौन से क्रोमोसोम अरेंज हो जाते हैं? यू हैव ब्लू वाले 23 maternal dyad chromosome, that is N2C. N represents maternal, 2C, because if you remember, S phase के बाद you have 4C. S phase के बाद you have 4C, of which maternal is 2C and paternal is 2C. So what does N represent? Maternal. And how many chromosomes are there? 23 chromosomes, 23 maternal. But they are dyad, so it is 2C. तो ये है 23 मैटरनल क्रोमोसोम्स एंड ये हो गए 23 पैटर्नल क्रोमोसोम्स. And where are they arranged along the equator? They are arranged along the equator. This is a metaphase. ये metaphase है. And centrioles have reached the opposite sides. Centrioles opposite side reach हो गए हैं. From centriole arise fibers which attach it to the cell membrane. Fibers which attach it to the cell membrane. Those fibers are called asters. Asters attach centriole to the cell membrane. By the way, do we remember centriole is found only in animal cells? Centriole plant cell में होता ही नहीं है. So these asters are found attached to cell membrane only in animal cell. Plant cell में there are no asters because there are no centrioles. The spindle fibers are attached from cell wall to cell wall. So no centrioles means no aster rays. So plant cell में you have asters attaching centriole to the cell membrane. And centriole to centriole you have supporting fibers which are called as interpolar fibers. Centriole to centriole supporting fibers are called interpolar fibers. ये interpolar fibers होते हैं. और बहुत ध्यान से do we remember ये है क्रोमोसोम और क्रोमोसोम में बीच में है सेंट्रोमियर. And on the sides are found proteins. What are those proteins on the sides called, if you remember? Kinetochore. Those proteins are called kinetochore. And from the kinetochore arise fibers which attach the chromosome to the centriole. To the centriole, chromosomes are arranged, and those fibers are called as chromosomal fibers. What are those fibers called? Chromosomal fibers. So once again, ये है chromosome, ये है kinetochore, बीच में है centriole, बीच में centriole है. And from the kinetochore arise fibers attaching it to the centriole. From the kinetochore arise fibers attaching it to the centriole. And what are those fibers called as? Those fibers are called as chromosomal fibers. Those fibers are called as chromosomal fibers. And तो आपके तीन फाइबर हो गए. तीन फाइबर हो गए. From centriole to cell membrane are called asters. Centriole to cell membrane are called asters. Centriole to centriole are called as interpolar fibers. Are called interpolar fiber. And kinetochore to the centriole are called chromosomal fiber. So we have chromosomal fiber. So kinetochore to centriole chromosomal fiber, centriole to cell membrane are asters, and centriole to centriole are called as interpolar fibers. So what is kinetochore to centriole called as? Chromosomal fiber. What is centriole to centriole called as? Interpolar fibers. And what is centriole to cell membrane called as? Aster fibers. By the way, asters are found only in animal cells. They are not found in plant cells. This is metaphase. Now the cell goes into the next phase called as the anaphase. Anaphase में what will happen? सबसे crucial. ये है आपके chromosomal fiber. Remember, chromosomal fibers are made of tubulin, actin-myosin, actomyosin, they start undergoing contraction. The chromosomal fibers start undergoing contraction, and because of contraction of chromosomal fibers, it will create a tremendous pull on the centromere. A lot of pull is done on the chromosome, और क्रोमोसोम के ऊपर काइनेटोकोर से पुल होता है, काइनेटोकोर से पुल होता है. All that pulling will act on the chromosome, और क्रोमोसोम के बीच में कौन था? Centromere. And because of pulling, the chromosomes made up of chromatids start moving to the opposite pole. यहां क्रोमोसोमल फाइबर पुल कर रहा है, ये क्रोमैटिड इधर पुल होगा. ये क्रोमोसोमल फाइबर इस तरह पुल कर रहा है, तो ये क्रोमैटिड इधर पुल होगा. राइट पुल, लेफ्ट पुल, राइट पुल, लेफ्ट पुल, and all the pressure will act on the centromere, which is connecting the two chromatids, and centromere will break. The centromere splits during the anaphase. Anaphase में centromere will split, and chromatids start getting pulled by chromosomal fibers to the opposite sides. तो एक क्रोमैटिड एक साइड जाएगा, दूसरा क्रोमैटिड दूसरी साइड जाएगा. So all the chromatids are now moving towards opposite pole. और यहां कौन है? सेंट्रोमियर. सेंट्रोमियर जो टूट चुका है, ये है सेंट्रोमियर जो टूट चुका है. और उसके पीछे पीछे कौन आ रहा है? क्रोमैटिड्स which are attached to it. Chromatids which are attached to it. So who is moving first towards the centriole? The centromere. और सेंट्रोमियर के पीछे पीछे कौन आ रहा है? क्रोमैटिड्स. And from the chromatids arise fibers which are called as interchromosomal fibers. तो दो क्रोमैटिड जो टूटने के बाद बनते हैं, what are those fibers called? Interchromosomal fibers, also called interzonal fibers. Interchromosomal fibers, also called interzonal fibers. And golden words, interchromosomal fibers undergo stretching. They stretch and they push chromatids. They push chromatids. They stretch and they push the chromatids. If you remember, chromosomal fibers are pulling chromatids. Chromosomal fibers are pulling chromatids. ये chromosomal fibers are pulling chromatids. They are pulling chromatids. And interchromosomal fibers are pushing chromatids. तो ध्यान से, who is pushing chromatids? Pushing chromatids or stretching is done by interchromosomal fibers or interzonal fibers. And what do chromosomal fibers do? Chromosomal fibers are pulling chromatids. They are pulling chromatids. So once again, chromosomal fiber pulls chromatids, and interchromosomal fibers pushes the chromatids, and chromatids go to the opposite poles. What is that phase called as? Anaphase. Golden words. Sir, what was the cell in metaphase 2N4C? It was 2N4C. We had N maternal and we had N paternal. So it was N and N. But how many chromatins were there? Four chromatins were there. So it was 2N 4C. But after centromere splits, what do we get here? N maternal. N maternal. And here we have N black, which is N paternal. N maternal, N paternal. But यहाँ पर भी N maternal blue, N paternal black. So it becomes 2N 2N. 2N remains same because you have maternal, paternal, maternal, paternal, blue, black. Similarly, here also you'll have blue, so maternal, black, so paternal. Here you'll have blue, maternal. Here you'll have black, paternal. So n plus n becomes 2n. वो तो सेम ही रहेगा क्योंकि maternal paternal दोनों साइड जाएगा. But how many C's we had? 4C. 4C was there. Now, as centromere splits, what will this become? C. And what will this become? C. So 4C will become C plus C, that is 2C. और यहाँ पर भी 4C has become 1C plus this is the other C. So C plus C equals 2C. So what has 2N 4C now become 2N 2C and 2N 2C? 2N 2C and 2N 2C. If you remember, what was the cell initially where it was a zygote? It was 2N 2C. There was N maternal, N paternal, that was N plus N. And how many chromatin threads were there? 2. And 2N 2C after S phase had become 2N 4C. But after anaphase, due to splitting of centromere, we are having N plus N, 2N, but 2C, N plus N, maternal, paternal, but 2C. So 2N, 2C, 2N, 2C. In other words, chromosome number remains the same. And this is mitosis where chromosome number will remain the same. So 2N4C has now become 2N2C and 2N2C, where chromosome number has remained the same. In which phase has that taken place? In the anaphase. Anaphase में ये हुआ है. And then each chromatid is now called as daughter chromosome. Each chromatid is called as a daughter chromosome. So each side will receive 23 maternal and 23 paternal chromatids. In other words, 23 maternal chromosome and 23 paternal chromosome. हाँ, तो ये जो क्रोमोसोम था, ये कौन सा था? डायड क्रोमोसोम. Dyad chromosome with identical sister chromatids. Dyad chromosomes, if you remember, these were dyad chromosome with identical sister chromatids. अब ये कौन हो गए हैं 46 मोनैड क्रोमोसोम, 46 मोनैड क्रोमोसोम, ये साइड भी क्या आ गया 46 मोनैड क्रोमोसोम। डायड हैज बिकम मोनैड एंड ये है सिस्टर क्रोमैटिड, ये है सिस्टर क्रोमैटिड व्हिच आर जेनेटिकली सेम, ये है सिस्टर क्रोमैटिड, ये है सिस्टर क्रोमैटिड जेनेटिकली सेम, ये है मैटरनल मैटरनल सेम सेम, ये है पैटर्नल पैटर्नल सेम सेम, and that is the way you have 2N 2C formed on either side. What is each chromatid now called as a daughter chromosome? So each side receives how many chromatids? 23 maternal chromatids, that is 23 maternal chromosome, and 23 paternal chromatids. Yeah. So 23 maternal and 23 paternal chromosomes will be formed. And that is anaphase. From anaphase, the cell goes into telophase. तो यह है anaphase. Anaphase से the cell will go into telophase. Telophase में the daughter chromosomes uncoil and undergo hydration to form chromatin network. Telophase is exactly opposite of prophase. Telophase is exactly opposite of prophase. What will happen? Daughter chromosomes uncoil and undergo hydration to form chromatin network. So chromatids will become super solenoid, will become solenoid, will become nucleosome fiber, bit, will become mid string, mid string, mid string, mid string, chromatin fiber. So it will go into original mid string stage, that is by hydration. So it will uncoil. Prophase में chromatin fiber had coiled to become chromatids and chromosome. After anaphase, splitting of centromere, each daughter chromosome chromatid will become by hydration and uncoiling chromatin fiber. Bead string, bead string, bead string, bead string, bead string. That is chromatin network is formed. Nuclear membrane begins to reappear and daughter nuclei are formed and spindle fibers disappear and nucleolus reappears. So if you remember, what will happen in telophase? In telophase, ये सब उल्टा हो जाएगा. न्यूक्लियर मेम्ब्रेन वापस आ जाएगा. न्यूक्लियर मेम्ब्रेन वापस आ जाएगा. The nucleolus will appear. Nucleolus will appear. Spindle fibers break apart. Spindle fibers जो बने थे एनाफेज और मेटाफेज में वो सब टूट जाते हैं. And centrioles go back near the nucleus. Centrioles periphery पे थे, they go back near the nucleus. So it is exactly opposite to that of prophase. Prophase में उल्टा हुआ था, centrioles had gone to the poles. Telophase में centrioles come back to the nucleus. Late prophase में न्यूक्लियर मेम्ब्रेन एंड न्यूक्लियोलस डिसअपियर हुए थे. इन केस ऑफ टीलोफेज न्यूक्लियर एनवेलप एंड न्यूक्लियोलस रीअपियर हो गए. स्पिंडल फाइबर बने थे. टीलोफेज में स्पिंडल फाइबर विल ब्रेक अपार्ट. तो प्रॉपर दो डॉटर न्यूक्लियोलाई फॉर्मेशन स्टार्ट हो जाता है. Two daughter nucleoli formation begins, and this is end of karyokinesis. Prophase, metaphase, anaphase, telophase, cell division, karyokinesis बंद हो जाता है, and in the telophase itself begins cytokinesis. Telophase में ही cytokinesis होता है. From the periphery there is something called as a cleavage furrow. A constriction in the cell membrane appears. Cell membrane में एक constriction आता है. in telophase itself. And let's see how proper cytokinesis takes place. In animal cells, furrowing of the plasma membrane deepens and daughter cells are formed, and this process is called cleavage. तो क्लिवेज फरो आता है, सेल मेम्ब्रेन में एक क्लिवेज फरो आता है, एंड क्लिवेज फरो विल कीप ऑन एक्सटेंडिंग and extending till what will happen, ये cleavage furrow extend होते ही रहता है, होते ही रहता है till cleavage furrow completely disappears and two daughter cells are formed. So cleavage furrow in the cell membrane from periphery to the center will divide the animal cell into two daughter cells. In case of a plant cell, something ulta will happen. Not from periphery to center, but from center to periphery is produced a cell plate. एक सेल प्लेट बनती है जो सेंटर से पेरिफेरी की तरफ जाती है, एंड दिस सेल प्लेट इज आल्सो कॉल्ड एज मिडल lamella, made of middle lamella, made of calcium pectate. कैल्शियम पेक्टेट नाम का एक केमिकल होता है which forms the middle lamella that forms the cell plate and divides one plant cell into two plant cells. So what happens in case of animal cells? A cleavage furrow from the periphery to the center divides and divides and divides one animal cell into two animal cells. In case of plants, a middle lamella, calcium pectate, also called cell plate, arises from the center, goes to the periphery, and divides one plant cell into two plant cells. Beautiful process, mitosis, very crucial. आगे हम लोग जब जेनेटिक्स करेंगे ये बहुत डिटेल में आने वाला है आपको सेल डिविजन चैप्टर. प्लीज प्रिपेयर दिस thoroughly well. I hope you understood this. This is mitosis. Next session we go into a very interesting concept called as meiosis.